Hello, welcome to Biology, the science study of life. Target tissues for insulin and glycogen. Insulin lowers blood glucose levels by stimulating virtually all body cells except those of the brain to take up glucose from the blood. Brain cells are unusual in being able to take up glucose without insulin. As a result, the brain has access to circulating fuel almost all the time. Insulin also decreases blood glucose by slowing glycogen breakdown in the liver and inhibiting the conversion of amino acids and glycerol from fats to glucose. The liver, skeletal muscles, and adipose tissues store large amounts of fuel and are especially important in bioenergetics. The liver and muscles store sugar as glycogen, whereas adipose tissue cells convert sugars to fats. The liver is a key fuel processing center because only liver cells are sensitive to glycogen. Normally, glycogen starts having an effect before blood glucose levels even drop below the set point. In fact, as soon as excess glucose is cleared from the blood, glycogen signals the liver cells to increase glycogen hydrolysis, convert amino acids and glycerol to glucose, and start slowly releasing glucose back into the circulation. The antagonistic effects of glycogen and insulin are vital to glucose homeostasis and thus to the precise management of both fuel storage and fuel consumption by body cells. The liver's ability to perform its vital roles in glucose homeostasis results from the metabolic versatility of its cells and its access to absorbed nutrients via the hepatic portal vessel, which carries blood directly from the small intestine to the liver. Diabetes mellitus, when the mechanisms of glucose homeostasis go awry, there are serious consequences. Diabetes mellitus, perhaps the best known endocrine disorder, is caused by a deficiency of insulin or decreased response to insulin in target tissues. There are two major types of diabetes mellitus with very different causes, but each is marked by high blood glucose. In people with diabetes, elevated blood glucose exceeds the reabsorption capacity of the kidneys, causing them to excrete glucose. This explains how or why the presence of sugar in urine is one test for diabetes. As glucose is concentrated in the urine, more water is excreted along with it, resulting in excessive volumes of urine and persistent thirst. Without sufficient glucose available to meet the needs of most body cells, fat becomes the main substrate of cellular respiration. In several cases, acidic metabolites formed during fat breakdown accumulate in the blood, threatening life by lowering blood pH. Type 1 diabetes mellitus, insulin-dependent diabetes, is an autoimmune disorder in which the immune system destroys the beta cells of the pancreas. Type 1 diabetes, which usually appears during childhood, destroys the person's ability to produce insulin. Treatment consists of insulin injections, usually several times daily. In the past, insulin for injections was extracted from animal pancreases, but now human insulin can be obtained from genetically engineered bacteria, a relatively expensive source Type 2 diabetes or second type diabetes mellitus, non-insulin dependent diabetes, is characterized either by a deficiency of insulin or more commonly by reduced responsiveness of target cells due to some change in insulin receptors. Although heredity can play a role in type 2 diabetes, research indicates that excess body weight and lack of exercise significantly increase the risk. This form of diabetes generally appears after age 40, but young people who are overweight and sedentary can also develop the disease. More than 90% of people with diabetes have type 2 or second type. Many can manage their blood glucose level with regular exercise and a healthy diet. Some require drug therapy. Adrenal hormones. Response to stress. Adrenal glands are adjacent to the kidneys in mammals. Each adrenal gland is actually made up of two glands with different cell types, functions, and embryonic origins. The adrenal cortex, the outer portion, and the adrenal medulla, the central portion, like the pituitary gland. Each adrenal gland is a fused endocrine and neuroendocrine gland. The adrenal cortex consists of two endocrine cells, whereas the secretory cells of the adrenal medulla derive from the neural crests or crest during embryonic development. Non-mammalian vertebrates have quite different arrangements of the same tissues. Catecholamines from the adrenal medulla. Suppose you sense danger or approach a stressful situation, such as speaking in public. Your heart beats faster and your skin develops goosebumps. What causes these reactions? They are part of the so-called fight or flight response stimulated by two hormones of the adrenal medulla, epinephrine, also known as adrenaline, and non-epinephrine, non-adrenaline. These hormones are members of a class of compounds, the catecholamines, that are synthesized from the amino acid tyrosine. Both epinephrine and non-epinephrine also function as neurotransmitters in the nervous system. Either positive or negative stress, ranging from extreme pleasure to a drop in temperature or to life-threatening danger, stimulates the adrenal medulla to secrete epinephrine and non-epinephrine. These hormones act directly on several target tissues and give the body a rapid bioenergetic boost. They increase the rate of glycogen breakdown in the liver and skeletal muscles, promote glucose release by liver cells, and stimulate the release of fatty acids from fat cells. The released glucose and fatty acids circulate in the blood and can be used by body cells as fuel. In addition to increasing the availability of energy sources, epinephrine and non-epinephrine exert profound effects on the cardiovascular and respiratory systems. For example, they increase both the rate and the stroke volume of the heartbeat and dilate the bronchioles in the lungs, actions that increase the rate of oxygen delivery to body cells. This is why doctors prescribe epinephrine as a heart stimulant and to open breathing tubes during asthma attacks. The catecholamines also cause smooth muscles of some blood vessels to contract and muscles of other vessels to relax, with an overall effect of shunting blood away from the skin, digestive organs, and kidneys, while increasing the blood supply to the heart, brain, and skeletal muscles. Epinephrine generally has a stringent effect on heart and metabolic rates, while the primary role of non-epinephrine is in sustaining blood pressure. As shown in this video, notice that In response to a stressful stimulus, nerve impulses from the hypothalamus travel to the adrenal medulla, where they target the release of epinephrine. Norepinephrine is released independently of epinephrine. The adrenal medulla hormones provide another example of a simple neurohormone pathway. Notice that this represent adrenal medulla secretes epinephrine and norepinephrine. Notice that, closer look, and notice also long term represent stress response effects of mineral notice retention of sodium ions and water, kidneys, increased blood volume and blood pressure, effects of glucocorticoids and mineral corticoids, for instance. And notice also protein and fats break down and converted to glucose, leading to increased blood glucose. Immune system may be suppressed. Case the notice also the in this case the neurosecretory cells are modified peripheral nerve cells rather than the hypothalamic neurosecretory cells that release hormones in the posterior pituitary. Steroid hormones from the adrenal cortex. Hormones from the adrenal cortex also function in the body's response to stress, but in contrast to the adrenal medulla, which reacts to nervous input, the adrenal cortex responds to endocrine signals. Stressful stimuli cause the hypothalamus to secrete a releasing hormone that stimulates the anterior pituitary to release the tropic hormone ACTH. When ACTH reaches the adrenal cortex via the blood stream, it stimulates the endocrine cells to synthesize and secrete a family of steroids called corticosteroids. In another case of negative feedback, elevated levels of corticosteroids in the blood suppress the secretion of ACTH. The two main types of corticosteroids in humans are the glucocorticoids, such as cortisol, and the mineral corticoids, such as aldosterone. Evidence is mounting that both glucocorticoids and mineral corticoids help maintain homeostasis when the body experiences stress over an extended period of time. The primary effect of glucocorticoids is an bioenergetics, specifically on glucose metabolism, augmenting the fuel mobilizing effects of glucagon from the pancreas. Glucocorticoids promote glucose synthesis from non-carbohydrate sources such as proteins, making glucose available as fuel. Glucocorticoids act on skeletal muscle, causing a breakdown of muscle proteins. The resulting carbon skeletons are transported to the liver and kidneys, where they are converted to glucose and released into the blood. The synthesis of glucose from muscle proteins is a homeostatic mechanism, providing circulating fuel when body activities require more than the liver can mobilize from its glucogen stores. It can also be part of a broader road of the glucocorticoids that of helping the body withstand long-term environmental challenge. Cortisol and other glucocorticoids also suppress certain components of the body's immune system because of their anti-inflammatory effect. Glucocorticoids have been used to treat inflammatory diseases such as arthritis. However, long-term glucocorticoid use can have serious side effects owing to their metabolic actions, and also can increase susceptibility to infection owing to their immune suppressive effects. For these reasons, non-steroidal anti-inflammatory drugs generally are preferred for treating chronic inflammatory conditions. Mineral glucocorticoids act principally on